Q&A sessions every week, just in case you have questions that you want to ask me. Those won't be mandatory. But if you have any questions about the course itself or any kind of content that you've seen, then I was going to do that. I guess I should just talk more about the course itself, I guess, rather than wait till 4 o'clock. So remember, it's the lab portion. So worth about 30% of the grade. And what we're going to do is do these labs, and the A&P labs can be done online. Matter of fact, last semester I had taught 204, the second half of A&P, and there's one section that had completely online labs for the whole semester. Hopefully we won't have to do that, but we'll see how it goes. And so remember, the lectures, however, are completely online. Those are video lectures that I'm making, and I'm uploading them to the YouTube site. You should be able to see the YouTube URL in the syllabus. You go to there and to view those. And I've already gotten through chapter 1 and chapter 4. Skipping chapter 2 and 3, those I'm not going to have lectures on. Those I have connect assignments. I'm going to connect assignments for those as a refresher for general chemistry and chemical reactions. And then and I'll even have I'm recording this lab in case people couldn't make this lab to put it up on YouTube, and you can see it again, or they can see it on YouTube. I will also occasionally open up the YouTube sessions for the group activities. We have about 7 or 8 group activities. These are problems, physiological problems that I usually used to pass out in lab, and we don't have a real actual lab each week, and sometimes you just pass out group activities. And I like to give the group activity and going into what are called breakout rooms for you to get together and answer these questions on the group activities. I'll be making group activity 1 available real soon. I should have made it available already. Okay, remember the exams are going to be completely online also, and they have to be done with something called Proctor Track. I don't know if you're familiar with Proctor Track, but it's a software that allows you to take exams at home. And I'll describe more about those now, in order to take an exam on Proctor Track, you have to take what's called an onboarding quiz that will do like face recognition and see your ID to make sure you are who you say you are. And you need to do that at least several days before the first exam, because there's a lag time between when it can process the proctoring exam and you can actually take an exam. It's going to be weekly online quizzes every week on the material that we cover that week. Those will be up in D2L. They'll start on a Thursday usually, go through Monday night, and those will not be on Proctor Track. Those will be just completely open resource. Right? And I'm going to probably allow second attempts on the exams, and they'll probably be open book as well. But I'll describe the exams, how they'll be set up later on when we do the first exam. So it's a quick and dirty example. There'll be connect assignments, like I said, for every chapter in the McGraw-Hill site. I call those homework. And they're not graded. Well, they're worth a certain number of points each one, but they're not particularly graded. They're just graded as long as you do them, and you do at least 50% of them, then you get the points really good because they question you in an adaptive way, and really they're good, great study tools, the Connect assignments. Okay. I don't know if I left out any. Does anybody have any questions? Anytime you got any questions, just come on and hit me with them. Also with the emails, you know, it's one of my major roles every semester is to answer emails, but particularly this semester when we're all remote, you know. So don't ever worry about sending me an email or asking me a question about anything. All right. Okay. So the way you get um, points in lab is doing, we have lab quizzes. So we're going to go over the microscope lab today. And then next week you'll have a online lab quiz on the microscope, worth 30 points. Each lab is worth 30 points. I think the last one on bone is worth 60 points. And I think also I dropped the lowest lab grade, I believe. We also have a couple of lab activities, one on endocrine and one on the autonomic nervous system. Hopefully, I don't know how I will be able to do those online. So I'm hoping we'll be on campus at that time. But I don't know. We'll see how things go. Well, whatever comes our way, we'll deal with it, and we'll make do and do the best we can. Right? So in these really strange times. All right. Okay. The microscope. I was going to have you come on the campus and look at the microscope. Oh, somebody was kicked out. Sometimes people get kicked out of the meeting. Let me readmit it. To look at the microscope for a couple reasons. In order to be successful with the microscope, you follow this procedure, and to be able to be successful and actually see things under the microscope. It's actually not a straightforward thing what you need to do. And also to protect the microscope. If you don't handle the microscope properly, you could damage it, and we didn't want to damage it. So I was going to bring in and give you some practice with the microscope with the first lab, and then the first lab quiz will be asking you questions about the microscope, and we'll get to those. Because next week, we'll be doing tissues. And in order to look at tissues, you got to use the microscope, right? So we will learn about the microscope, the parts of the microscope, how to use the microscope, how to be successful with the microscope, some terminology dealing with the optics of the microscope. And excuse me 1 second. Yes. We had a student in this class that was coming actually on campus, not realizing that we're doing things 100% online. Okay. So we're going to make believe we're going to be using the microscope, but we won't actually be using the microscope. We still got to know about the microscope. And and we'll go over to what you need to know for the lab quiz. Whoops, another person coming in. Lab quiz next week on the microscope. And then when we do the tissue slide, tissue lab, I'll just show you pictures of these tissues taken off the web and talk about each one, the ones you need to know. And then the lab quiz will be those same pictures from the web, you know, on the tissues. So this is how we'll see how it's going to work. Now, you should have a document called Microscope Lab that's available in D2L. Let me actually show it to you. I can do something here. Called screen sharing. I do the same thing on the video lectures. I have a PowerPoint for that chapter, and I pull it up, and you can see it on your screen. And then over on their side, you can see my face in the background. And then I run through the PowerPoint by screen sharing. So I'm going to screen share now and show you the microscope document. Okay, and so now you're over here. You should be able to see the microscope document, right? Everybody can see it. So I'm going to go over this and tell you what you need to know. And there's a general overview of the microscope and history of the microscope, but I'm not going to ask you questions about history of the microscope. Whoops, wait a minute, I just lost it. Where is it? Oh, here I can go down. And it talks a little bit about cells, and actually, I'm not going to go over this. This is actually in the next chapter, chapter 4, is all about cells. And there's going to be 2 video parts of it, one on cells themselves, and I already put that up. And then there's going to be a couple video lectures on a little bit on genes and genetics. And this is all on chapter. So I'm not going to go over eukaryotic cells and this stuff right now. Now, parts of the microscope. I just realized this. So here's a microscope right here. And you're going to need to know parts of the microscope, and there'll be, on the quiz, there'll be a picture of a microscope with arrows. And you'll need to tell me what part that is. Okay. But this microscope, I just realized this, is different than a microscope we use. It has some of the same parts in the same places, but it's not actually the microscope we use. So I'm going to, I just took a picture of the microscope that we use. I'm going to pull that up and screen share that. I'm also going to be putting this in D2L because that's the microscope. It's sitting on my desk. I'm in my office right now, and just a few minutes ago, I took this picture and uploaded it. And I was proud of myself by doing this because I'm technologically challenged, and I was able to take the picture with my cell phone and send it to my email, and do that in only a couple of minutes when it took me like hours last semester to figure that out. Okay. So there's the microscope, and this is called a compound microscope because you actually look through the object on the slide. Light's coming right through it, and you see right through it. So and so it comes up. Oh, now you can see the pointer. The light source is down here. This is a light switch right here, and I was always in class would come in and say, make sure you plug it in, turn the light switch on, and make sure the light comes on, because the microscope is useless without the light coming on. Okay, light comes up here, comes up through here, into this lens here, which is called an objective lens. There are 4 objective lenses, and they're on a revolving nose piece. You can select each one by clicking it in. They're color-coded. This one's set up to be red, which is called scanning, which is 4x, 4 power. There's another one that's yellow, that's called low power, that's 10x. There's another one blue called high power 40x, and there's another one right here that we don't actually use anymore called oil immersion lens, and that's 100x, and it's black or white. If your light goes up through here, into these oculars. This is an ocular, this is a second lens. So it's a two-lens system. Through the objective lens and through the ocular lens. The ocular lens is 10x. And there's two of them, so it's a binocular microscope. And so the total amount, what's called total magnification, the total amount that you expand your subject to, is just a simple product of the power of the objective lens times 10, because this is always the same. The oculars are always 10. So if it's set up with the scanning, it'd be 40, 10 times 4. If it's set up with the oil immersion, it'd be 100, 10 times 10. I, 10 times 40 or 400, and if it was oil immersion, 10 times 100 or 1,000. So that's just called total magnification. It's one of the terms you need to know. I wonder why my face is so dark. Anyway, okay. So here is the called the stage. Oh, I should have showed you the other side of the microscope. I don't know if I can do that. We can just hold it up to you and show you. Anyway, on this side, here's the stage. And if you see two little arms there, that's called the stage, the slide holder. And this is where you put the slide on. You put the slide on the stage flat, slide it between these two arms. And there's a couple little flanges over on the left. You squeeze the flange and it moves one arm out. You put the slide all the way back, you release the flange and it grips the slide. And then you move the slide with two wheels. Let me stop screen sharing for a second. Maybe I can, I hope this is gonna work. Wait a minute here. Maybe too dark. Can I get some more light around it? Here's the microscope. It's too dark. All right. What I can show you is, I go back to the other, Interestingly enough, this other microscope has two wheels that are down here. Called the coarse focusing and fine focusing? No. On that microscope, there are two wheels in the exact same position, but they move the slide holder. They move it forwards and backwards and left to right. So this is how you navigate the slide, how you move to look at different areas of the slide, right? So there on that side of the, I should have taken two pictures. Okay, we'll take two pictures and I'll put both pictures up on D2L to show you the other side of the microscope. I forgot about that, that the adjusters of the slide holder were only on the left side of the microscope. All right, or on one side of the microscope. Okay. Let me go back to our microscope. Okay, so then a big thing is being able to obviously focus on the object that's on the slide. And usually what we do, let's go through the procedure you use to look at anything on the microscope. Doesn't matter what it is. You put the slide on the slide, how can I figure the name? Stage. You put the slide on the stage. You put it between the arms. You grip it with the holder. You move the slide so that the middle of the slide is in the beam of light coming up from below. And then you switch to low power. You always start on low power. Whatever you're looking at, doesn't matter. Start on low power. And then what you do is you turn, there's two knobs here, nested knobs. A bigger knob here, a smaller knob here. This is coarse focusing and fine focusing. And how you start off is you turn the coarse focusing, as long as you're on low power, you turn coarse focusing so the stage is moved up as high as it can be. See, the focusing knobs move this stage down and up. The coarse focusing moves it a large amount up and down. The fine focusing moves it tiny amounts up and down. So you want to start with the stage as high as possible. And this is called working distance. Working distance is the distance between the stage and the bottom of the objective lens, right? And you want to start a working distance with low power for everything. And then when you have that, you turn the coarse focusing knob until you get your object in view. Right? I mean, not in view, but in focus. Right? Then when you have your object in focus, generally, the things we have to look at are so small that we have to go to a higher power to see them clearly. So we have to go to high power. So then what you do is you take a part of your object and you move it to dead center of what's called the field of view. The circle that you see, the white circle you see when you look through a microscope is called the FOV, the field of view. And you move some part of the object into the middle, dead center of the field of view. And then you click in, you turn the rotating knob and you click in the high power blue. And then you touch the fine focusing knob, not the coarse focusing knob. There's something I would really emphasize in class. You never touch the coarse focusing knob with when you're using high power, because the higher the power of the objective lens, the longer it is. So the smaller the working distance. And so when you're working on high power, you have a really tiny working distance. And if you use the coarse focusing adjustment, you could crash the slide right into the lens if you weren't careful. That's why you only use fine focusing under high. You can use actually fine or coarse under low, but only use fine under high. Now, something that will happen is when you go to high power from low power, the amount of light coming into your eye will decrease because that working distance is smaller. So you have to learn techniques to increase the amount of light that comes into your eye. And there's two ways you can do this. One way, you can just turn up the on-off switch. But the better way is using a little arm that's in front here. And again, hard to see it. It's in front here that turns something called the diaphragm. It's part of this black thing underneath here called the substage condenser. We generally don't use a substage condenser, but in a substage condenser is this diaphragm. And it's like the f-stop in a single lens reflex camera. Makes a big opening or a small opening. And the bigger the opening, the more light comes into your eye, the smaller the opening, the less light comes into your eye. And so sometimes you have to open up the diaphragm a little bit going into high power because there's less light coming into your eye. Although they're an important part of the diaphragm, is also helping with focusing, because there's an optical property that the smaller the opening you look through, the greater what's called the depth of field. When you look at something, and you're focusing on something with your eyes, that thing is in focus, right? There's something, and there's a certain amount of stuff in front of that object that's in focus, and a certain amount of stuff behind that that's in focus. And the rest is a soft blur. That's called depth of field. And the smaller the opening you look through, the greater the depth of field. So this is something that any photographer that uses a single lens reflex knows. The higher the f-stop, the greater your depth of field. So this helps with focusing as well. If you use you look through a smaller opening. Okay. Yeah, I think that's it for the parts of the microscope. You may have any questions, anytime, don't worry about interrupting me about a question about anything. Well, there's one more wheel here that moves the substage condenser up and down, but we generally don't use that. We don't use that. Okay. Let me go back to the our okay. So use those pictures, not this slide, for the parts of the microscope. Okay. So microscope terminology. You need to know a few definitions of terms. Magnification, and it really should be called total magnification. Well, magnification is how much each lens magnifies it. Total magnification is always the product, as I mentioned, of the objective lens times the ocular, and the ocular is always 10. So if you look at magnification of ocular, it's always 10. Magnification objective, low power 10, high power 40, for immersion 100. So total magnification down here again would be 100, 400, 1000. So if I give you the magnification objective, you could tell me what the total magnification is. Just multiply it by 10. All right. Resolution, ability to differentiate detail, to see fine detail. And the only relationship you got to worry about there is the higher the magnification, the greater the resolution. All right. So there should be some relationships here that I could ask you about. And that's one relationship. The higher the magnification, the greater the resolution. Okay, contrast, really important thing. Contrast is the ability to see something against its background. And that's always a problem with a compound microscope, because we're generally looking at very tiny things, and these tiny things are generally either not colored or transparent. They're transparent, the light's going right through them. So here's the problem. We want to look at a colorless thing or transparent thing on a white background. So we got a contrast problem. It's like trying to see a polar bear in a snowstorm. So we always have to use techniques to increase contrast. And there's a couple ways to increase contrast. One of which is to dye something, stain it. And we'll be using stains in lab. For example, we do a cheek skin cell, a cheek cell lab where these cells are flat and they're transparent and they're not colored. Well, we put this yellow dye, either a yellow dye or actually a blue dye, on it and make them stand out in the background. So you can stain stuff. Another way is very simply to reduce the amount of light that's coming into your eye. If you want to look at something that's not colored or transparent, reduce the amount of light. This is a common mistake students make, is they let a lot of light into their eyes, and it washes out these these transparent things. And it's really amazing if you just see like blurs and you just turn the light down, turn the diaphragm so there's less and less light, and things just come out and you can see them. So that's two things. So here's the thing: if the thing you're looking at is already stained, then you want to use a lot of light to look at it. If it's not stained, you want to use less light to look at it. Okay. Numerical aperture. Don't worry about that. I'm not going to ask you anything about numerical aperture. Parfocal, this means that this is why if you see something under low power and in your field of view, and let's say it's off to the side, you can see it off to the side, you're the edge of the field of view, and you want to see it under high power. Well, if you just focused in on it and left it where it was, and you went to high power, you would miss it. It wouldn't be in the field of view because there's another relationship. The higher the magnifying power, the smaller the diameter of the field of view. So if you leave your subject over to the side of the field of view under low power and you switch to high power, you're probably going to miss it. And if you miss it, you lose it under high power, you will pretty much never find it. You can't find things under high power. It's almost impossible. Even with really accomplished microscopists, they can't find stuff under high power. So what you have to do is move, part of your object from over the side here and put it dead center. That's why I said move something to dead center. And then when you click in high power, a part of it will be in the field of view. It'll be blurry, and you can turn to fine focusing and you'll get it. So if you lose it between low and high, you got to go back to low, center it, and try it again, because you're just not going to find it under high power. Okay. That's called a parfocal microscope. Okay. Working distance, I measured it, I defined it, where it defines it slightly different here. This is between the objective and the slide. I said this is between the objective and the stage. Doesn't matter either way you want to define it. And remember, working distance is greater with lower magnification. Here's the relationship. Okay. Field of view, we mentioned that the higher the magnification, the smaller the field of view. And so if you go to higher magnification, you see greater resolution, but you see less of your subject, of your object, because the field of view has been reduced. All right. Phase contrast, don't worry about phase contrast microscope. You don't have to worry about that. A smear, we don't have to really, but, well, a smear is just making your own slide. In lab, we would have looked at two types of slides. One, pre-made slides that were made by some company, and we bought them, and we look at them, and then we put them back. But then there's ones we make ourselves. And these are called smears, and also called wet mounts. Like if you do it for your cheek cells, you scrape the inside of your cheek, make a smear in the middle of the slide in a small drop of water, take a cover slip and drop it over that. And so you make your own slide, and you can look at your own cells under a microscope using your own slide. And making that is called a smear. And usually we use biological stains to, we put it on the slide in the smear, and we drop stain onto the smear. We drop the cover slip on to see it. Okay, index refraction, I'm not going to ask you anything about index refraction. That just means that the light bends when it goes from one medium, like air, into another medium, like water. Okay, Okay, so let's go over the operating, again, the operating the microscope. Um, if you go pick up the microscope, you pick it up with two hands, just to make sure you don't drop it. There's no covers on microscopes anymore. 3, you gotta plug the microscope in, see the light works. And then you start with, number 4 is you start with low power. You always start with low power. Here are these same procedures that I already talked about: putting the slide on stage, putting it in the slide holder, moving it to the middle of the beam of light. Oh, and not using the coarse focusing, not using the slide holder controls. You move the slide to the middle. And then 7, look to the eyepiece, adjust the light so it's comfortable to your eye. And also, the 2 oculars are adjustable to set up for your particular eye width, the distance between your eyes. Okay, and then you focus in. To the object. You can use coarse focusing for this, and then you increase the magnification by rotating in the high power objective. Once you've moved a part of your image that you want to see dead center, so that when you move to high power, you'll see something. All right. And we won't be using your immersion lenses. Okay. Well, let's go over what we would have done in a lab, and we actually had it on campus lab. The first thing you look at would be a letter E. Real exciting. And when you look at the letter E under the microscope, you'll notice that it is flipped upside down and backwards. It's upside down and flipped left to right. And this doesn't mean your microscope is broken. This is an optical property of a 2-lens system. That when you look through an object with 2 lenses, it flips it upside down and reverses left right. So it's called an upside down mirror image. Because remember, if you look in the mirror, right becomes left and left becomes right, correct? Well, let's make believe you also, when you look in the mirror, your head's down here and your feet are up here. That's what you're looking at. And so it's something you can't get around. It's an optical property. You just have to deal with it. So later on, it has a little where was that? Oh, down here. This chart, it's just if you want to look at the right hand side of your object, you have to go left. If you want to look at the lower part of your object, you have to go up. You always have to go in the reverse direction, you think. And it's confusing at first, and it takes a while to get used to. But like I said, it's something you just have to deal with. Right? So, as a matter of fact, I always say in the lab also that it's an interesting physiological note that we'll get to when we get it to the senses, that your eyes work in exactly the same way. It's a 2-lens system. The first lens is your cornea. The second lens is actually called a lens behind your iris. And so your world is projected into the back of your eye, under your retina, upside down and reverse left to right. So the obvious question is, how can you see the world right side up? What's happening? Well, you ever think of what's called the visual projection pathway. That information from your eyes goes right back here to the occipital lobe. And those little gray cells turn things around forward, right? So that you see the world right side up and correct left to right, even though the image on the back of your eye is upside down and reversed. Okay. This taking the letter E from the slide tells you the exact same thing what you normally do with anything. You start with you put it in slide holder, you go to low power, you take a dark part of the E and you put it dead center and you go to high. And of course, under high, you won't see the whole E. You just see part of it, right? Because the field of view is much smaller. Okay. Then what we would also do is actually determining the size of the field of view. And so you'd get this slide with a little piece of a metric ruler. Oh, wait a minute. My tea. Sleeping too long. That's a green tea here. Okay. Okay. Get this metric ruler slide. And you have these bars that are millimeter bars. And I would have you look at this under low power. Line up the bars and count how many millimeters it takes to go from the left side of the field of view to the right side of the field of view. And with low power, that turns out to be 2. Almost everybody agrees it's around 2. It's a little off, but it's close to 2. And we're just looking for, you know, ballpark figures here. So that's 2 millimeters. But the problem is, millimeter is too big of a unit. It's a really small unit as far as, you know, your eye is concerned. But in the microscope, it's a huge unit. So we have to divide it into 1,000 into what's called micrometers. And so here's this 2 talks about how you convert millimeters to micrometers. So since there's 1,000 micrometers in every millimeter, if the diameter of the field of view with the low power objective is 2 millimeters, it's 2,000 micrometers, right? Okay, so you physically measure the diameter of the field of view under low power. Then you want to get the diameter of field of view under high power. Well, first of all, why do we even worry about this? Why do we care about what the diameter of the field of view is under the high power objective? It's to measure the size of things you see under the microscope, like cells. I want to know how big is this cell? Well, you see a cell in a field of view and there's nothing to compare it with, you know. You got to have a comparison to get an idea of how big it is. So if you know the diameter of the field of view and you can see the cell in that field of view, then you can get an idea of how big it is, you know, a guesstimate of how big it is. So here's but the problem is you can't measure physically using a ruler the diameter of the field of view under the high power objective because it's too small. So you have to calculate it because of a certain relationship between the magnification of an objective and its diameter field of view. Shows you that in a mixed equation. Magnification of the low power objective times the diameter of the low power objective is a constant. Matter of fact, it's always a constant. If you magnify, if you take the magnification of 10x objective and you multiply it by its diameter field of view, it's a constant. It's always a constant. It's the same for every single objective. And what is that constant? That constant is 20,000. Because magnification of low power here is 10. diameter of low power field of view is 2000. So 10 times 2000 is 20,000. Oh, wait a minute. Somebody wants to come in. The student who was just here. Hang on a second. I want to send her an invite. Wait, I think I have to get out of this first. Okay. Then let me send. So who is that? Okay, how's your? Okay, let me send her an invite. Oh no. Oh wait, let me copy it. Oops. Copy. All right. And then let me send it to her. Okay, sorry about that. When she gets in the waiting room, I should get a notification. Okay, so let me get back to this. So if you, like I say, you multiply the magnification objective times its diameter field of view, it's always the same. So you take it, so you measure it for the low power, you divide into that the magnification at the higher power, 40. So it'd be 20,000 divided by 40, which turns out to be like 2,000 divided by 4, which is 500. You see that? So we calculate the diameter field of view for the, oh, there she is. We calculate the diameter field of view for the high power objective by using that relationship. Right? So you gotta know how this works, because I could give you that under the on the quiz. I could say, okay, here's the diameter field of view under this power objective. If we had this power objective over here, what would be its diameter field of view? So it might be a little bit of math on the next lab quiz. I'm not saying it will be, but where's the? Oh yeah, in this part here, we'll be examining different types of cells, and we don't have to worry about that because, well, what I would be doing would be asking to look at a plant or an animal cell. This is how we do it, but you have to worry about these things because we're not actually going to do this. And here under the results, we would write down how the E looks under low and how it looks under high. And we've already done this chart here. You know, well, this is like really kindergarten, working distance. Under 10x, it's furthest away. Under 40x, it's middle. Under, 100x, it's closest. And total magnification, we already filled this out. Is multiply power of the objective times 10. All right. Okay, as you go from low power to high power magnification, does the image get lighter or darker? Well, it gets darker. Why? The working distance gets smaller. So what can you do to increase the amount of light? Two things. Move the diaphragm, that's the best thing, or turn the on-off switch, and that will increase or decrease light. Okay. Okay. Okay, type of stain, we have to, there's something, we had a crystal violet was the stain we used. Okay, okay, here, okay, and why was, well, we use iodine and crystal violet, and they're both stains. And why we use them? Increase contrast. And again, you don't have to worry about phase contrast microscope. Don't worry about that. And crystal violet used to increase contrast. And the last thing is, okay, if you wanted to know how big a cell was, and remember, this is why we were measuring the diameter field of view in the high power objective, which turns out to be 500 micrometers. Well, we would want to know, how big are these cells? And the way you do that is it worked the best with the plants, because we look at onion root tip, and you'd have all these rectangular cells that extend all the way across the field of view. So from here to here is 500 if you're using high power. Using low power is 2,000, but we usually use high power. So if there was a cell that extended from here to there, how long would that cell be? 500. If there were only two cells, if you counted across the number of cells at the thickest part of the circle, there was one cell, two cells. How big would each one of those cells be? How long would they be? 250 micrometers. How do you figure that out? Well, there's two cells across. So you divide the diameter field of view by the number of cells across. Two cells. If there were four cells across, it'd be 125 micrometers. 10 cells across, each one would be 50 micrometers. See what I mean? That's how you determine if you have cells that extend all the way across. And we would do this both with plants and animals, because we'd have you look at this slide called mitosis in whitefish blastula. And these are circular cells. If you go to high power, if you go to low power, they don't extend all the way across. But a high power, they extend all the way across, and you can count them and divide 500 by the number of cells across. And you got the average size of one of those cells. And it's not always, an even number. It might be 4.5 or 3 3/4 or 5 and a quarter or something like that. Whatever it is, just divide that number into the diameter field of view. Okay. So I think that's pretty much it of the things you can expect on the lab quiz. Like I said, I'm going to take another couple pictures of the microscope and put them up in D2L, because this is the microscope we'll be using, not this picture that's in the on our lab papers here. I didn't realize that we were using a different type of microscope. Okay. So, maybe I went a little bit fast, so I don't know if does anybody have any questions, any problems, any kind of concerns here?
So do we currently log on both Mondays and Wednesdays? And then if we go back on campus, you'll let us know if we're Monday or Wednesday?
Yeah, I figure online, we don't have to split it into 2 days, although we could, because everybody can fit in a Zoom meeting. So I was thinking we just do like next week. We since Monday was a holiday this week, so we couldn't use Monday. So Monday won't be a holiday next week. So I figure we'll do the same lab. We'll do a lab at 2:30 on Monday, and then on Wednesday we'll open it up again for anybody who has any questions about the lab. You see, and then the following week we'll have the lab quiz. Now, if we get back onto campus, like I said, we can only fit 11 students in the lab at a time. So I have to divide the lab up into 2 parts. And what I would do was assign half the students to take their lab on Monday, half the students take their lab on Wednesday. And I would usually do that by the letter of your last name. Like the first half of the alphabet goes on one day and the second half of the alphabet goes on the other day. But you actually choose a day if you want. If your work schedule or something works better with a Monday or Wednesday, just let me know and I can put you into either day. And then you'd only take a lab on that one day. So eventually I hope that we'll get to that and actually have on-campus labs. See how that would work? Yeah, so next week we'll all get together on Monday and do the next lab. And yeah, in a normal semester, labs will be 2 3/4 hours long, but dividing them into 2, they're 75 minutes long. So they're a lot of studying for a lab has to be done outside of lab because there's barely enough time in 75 minutes to take a lab quiz. And then for me to present you with the new stuff you got to learn for next week's lab quiz, and usually it would be, on models or preserved materials. Like I'll come to lab and tell you what you need to know on the models or on the preserved materials for the quiz the following week. And there won't be time enough for you to study them in lab really. So that's why a lot of, online and off-campus studying will be required. Oh, and you need to, if you have a chance to go in and take a safety quiz. Usually in the first lab, you should do safety contracts to give you safety information. And even though it doesn't apply to you on an online lab, it'll eventually apply to you if we get on campus. So instead of doing the safety contracts, now we have a safety quiz that you should do it's not a graded quiz or anything, just something to let you know about the safety proceedings. And now we have new safety proceedings, with social distancing and mask wearing. You got to wear a mask. You also got to pass the what's that called? The eco GoEvo PPA test, a little quiz about whether you've been exposed to COVID or not. And you can do it on your cell phone if you get the app, or you can do it on the computer as soon as you walk in the building, before you ever come into the building again. I do it every day because I come into the building just about every day. So I have to fill it out every day and send it to my supervisor to tell her that, it's okay for me to come into the building. So. And again, once we stop the meeting, I'll stop recording. This will be converted. I'll move it, and I'll upload it to YouTube. So if you want to watch this again, I may go on a little fast in certain areas. You want to see it again, it's going to be available in YouTube. Right. So in a couple minutes, I'll in the meeting unless in the lab unless we have any more questions. Well, particularly about the student came in really late, who came to my office. You should really go to YouTube and view YouTube because you missed most of the lab, so that'll help you particularly. Oh, and like I said, the group activities. I'll like I said, some of the labs are going to be group activities because I used to hand them out in a lab, and and um, and I like to do the group activities as a group and go into breakout rooms, but you can do them individually as well. Most of them last semester were done individually, but I'll explain how they work once um, and some of you've had me before as an instructor, and you know how they work, but I'll explain later on how they work. Make sure. Okay. Okay, like I said, I'll end it in a minute if no more questions. And if I do end it and you all of a sudden you think of a question that you wanted to ask, don't hesitate to email me. I mean, that's one of my major functions now is answering emails. And I'll try to not to miss your email and get it back to you as soon as I see it. And also, if you remember the question, I'm going to be doing another Zoom meeting in 25 minutes. And for other students who hadn't come in, and so if you can always come in and join that one too if you remember something you wanted to ask. So we're doing a lot of Zoom meetings. All right, so everybody's okay? Okay, well, okay. Well, good to see you, and let's hope we have a good semester and everything. And like I said, hang in there and be safe. And I'll see you later. All right, bye bye.